Hey everybody, welcome back to lecture 2. So now we're going to talk about some other microscopes. We talked about bright field microscopes in our last lecture video. So dark field microscopes are microscopes that produce a bright object against a dark background. And so the object itself stands out because the background is dark, whereas in the bright field microscopy, the object is dark and the background is bright. This is actually a mechanism when you're trying to see organisms that you can't stain. This is one mechanism for looking at those organisms, is using a dark field microscope. Phase contrast is a special type of microscope that allows you to look at specimens when they're not stained at all and still see a difference in different areas. So each of these figures, I should tell you, from the first figure to the second figure, these are all the same exact organism. This is an amoeba. And so you can see just how they look under different microscopes. So phase contrast, you can see different, it almost looks like you can see thicker regions of the cell and then thinner regions, like up here looks thicker, down here looks a lot thinner, where there's probably that pseudopod forming. And then we have electron microscopes. Electron microscopes are the strongest of our microscopes. They can magnify things over 100,000 times. They use electromagnetic lenses, they use electrons, and they use fluorescent screens to replace the glass lenses that we typically use, visible light and the eyes. So the image is photographed and then is placed on a computer screen, and then you can color it artificially. So wavelength of electrons are approximately 1,000 times shorter than light rays. And so we can see we have a much greater resolving power because of this. So 0.2 micrometers to 0.3 nanometers. So that's a huge difference in resolving power. When you're using electron microscopes, the lenses and the specimen have to be in a vacuum because air would interfere. You cannot look at living specimens. So if you're trying to look at something that's living, you'd have to use a compound microscope, like a light microscope or a dark field microscope. The two major ones that we have are your transmission electron microscope and then the scanning electron microscope. This is a figure of using a transmission electron microscope. So you can see that cell structure, the darker region on the outside is associated with a denser amount of tissue. You can see internal structures to some extent. So you can see like little vacuoles down here. You can see probably what the DNA looked like in there. They use freeze fracturing to reveal the shape of structures, or they have, but there are new methods that do less damage to the actual cell structures, cryo-electron microscopy and cryo-electron tomography, which is what you're seeing here. Scanning electron microscope then uses a beam of electrons that scans the surface of the cell, so you can really see the surface of the cell structure or the surface of the image that you're looking at. You don't see inside though, you only see the outside, so it's a 3D outer effect. And so if you're looking at all the microscopes, I hate it when this happens. So that says the instrument. It's covered up. I apologize for that. Mechanism and comment. And so here are the different microscopes, light microscopes that we talked about. Here's the mechanism and comments about them. Here we have some other light microscopes that I did not talk about. I only talk about certain ones, the most common ones that we use a lot. But you can read about these other microscopes as well. And then the transmission and scanning electron microscopes. I did not talk about the scanning probe microscope either, but you have that information right here. This next section is all about how we prepare our specimens so that we can use them under a light microscope. And so you can do a wet mount of a specimen, or you can do a dry mount. So wet mounts are used when you're trying to look at something. A lot of times we use wet mounts if we're looking at, say, pond water, so that we can see something underneath. Or say we want to look at what a drop of blood looks like in salt water. Then you might put a drop of salt water with a drop of blood, and then you could watch what happens to those cells. Whenever you use a wet mount, you use a cover slip. A cover slip is a tiny piece of glass, a very, very thin piece of glass that goes over the fluid. So you're using a very small amount of fluid, and then you're putting a cover slip to keep the lenses from getting wet. So this is an effective mechanism for looking at certain things, but not always effective if the materials we're looking at are colorless. And so in microbiology, we tend to make bacterial smears. And when you do this, you're going to dry the slide and fix the specimen onto the slide before you actually stain it. And so that's what you're seeing down here. You're seeing some microbes are being put onto the slide. The slide is then drying, and then we heat it by running it through a Bunsen burner flame to make sure that it is fixed and won't move, and that kills the bacteria as well. Then we stain it and look at it under a microscope. So there are multiple stain techniques. We'll talk about the simple stain. We'll talk about these different stains, but the ones that we typically learn in our lab are the simple stain, a negative stain, which isn't written on here, but it's another form of simple stain, a gram stain, an acid-fast stain, as well as endospore stain. We do capsule stain. A couple stains that we don't tend to do in our lab are flagella stain and fluorescent dyes. If you work in a hospital lab, you may use fluorescent dyes and fluorescence microscopy to identify pathogens, because we use this in an immunofluorescence test to see if there are certain types of antibodies in your system. All right, I'm going to stop here, and we'll talk about the different staining techniques in our next video, okay? Bye.